Hi, my name is Fabian. In this work, stimuli-responsive porphyrin containing fluorescent acrylate hydrogels were prepared by frontal polymerization.
Hi, my name is Mireille. Frontal polymerization is a simple, rapid, efficient, and green methodology for the obtention of different polymeric materials such as resin, nanocomposite, interpenetrating polymer networks, and hydrogels. It consists of a radical polymerization with a radical initiator promoted by an energy source in form of heat, UV light, infrared laser, ultrasound, plasma, etc. A polymerization self-propagating front disseminates throughout the monomer solution.
Hydrogels are polymers with remarkable water absorption capacities. They can be produced from natural sources or from biocompatible components such as saccharides, proteins, amino acids, polycaprolactone, polylactic acids, polyethers, etc., and can be formed reversibly by non-covalent interactions or irreversibly by covalent linkages.
Hydrogels are used for different applications in medicine, biology, pharmacology, material science, catalysis, etc. In the field of pharmacology, hydrogels can be used as drug delivery scaffold in photodynamic therapy and in fluorescence microscopy as biocompatible fluorescent probes.
The absorption and release of a drug depends on the physical and mechanical properties of the hydrogel, as well as on environmental factors. The stimuli-responsive hydrogel is advantageous for harnessing drug release in specific conditions of temperature, pH, and ion concentration. Frontal polymerization was used to obtain covalent acrylate hydrogels containing tetraphenylporphyrin as chromophore to endow them with fluorescent properties. We have synthesized the tetraphenylporphyrinyl tetraethylene glycol methacrylate monomer, TPP-MAM, and reacted in different amounts with two mixtures of diethylene glycol ethylacrylate and polyethylene glycol methacrylate in the presence of a radical initiator, APSO.
The obtained hydrogel series PD50 and PD10 were characterized by FTIR and thermal analysis, TGA and DSST. The composition of both series did not significantly affect the thermal properties and glass transition temperatures. The same was true for the hydrogels with TPP-MAM present in the polymeric matrix.
The swelling ability was studied under different temperature, pH values, and sodium chloride concentrations. The hydrogel swelling abilities were influenced by such stimuli. The observed effects were more evident in the PD10 series.
The observed fluorescence emission of TPP-MAM in solution was detected in solid state for all the TPP-MAM containing fluorescent hydrogels. Scanning electron microscopy revealed a rough surface, a morphological characteristic present for effective water absorption.
In this work, we have used frontal polymerization as an efficient, fast, and green methodology to obtain stimuli-responsive TPP-MAM fluorescent acrylate hydrogels. On the one hand, the monomer composition had no important difference in the thermal properties of the hydrogels. On the other hand, a more marked swelling response upon stimuli was observed for the PD10 hydrogel series, with higher diethylene glycol ethylacrylate ratio. As ongoing work, our hydrogels will be evaluated as photodynamic therapy and drug delivery candidates. Thank you.
Thank you!